Hello everyone, this is group 26 and our project is development of a non-invasive point of care malaria diagnostic utilizing a malaria-based self-free toehold switch biosensor. So our presentation is made up of mainly 4 parts, and these are, next slide. We have the introduction, objectives, workflow, and the conclusion. So on the background, which is part of the introduction, the study was done by the World Health Organization in 2022. It was published in 2023, whereby it was found out that 249 million malaria cases were found in 85 malaria endemic countries. And so we have to develop an essential and effective malaria management tool. Maybe we have to make sure we can diagnose it very well. And the traditional diagnostic tools that have been available are microscopy, maybe also PCR, and also the rapid diagnostic tests that is the RDTs. And the RDTs have been well favored because of their simplicity, and they are used mainly by the travelers and even in the field-based researchers. But in the study was done by Magne in 2024, and it was found out that there are deletions occurring in the Plasmodium falciparum, and there are deletions in histidine-rich protein, which makes it very hard to diagnose malaria because we get false negative results. So the problem statement, it includes the lack of specific and sensitive diagnostic tools, and also the mutation of the histidine-rich protein 2 from the falciparum, as I told you, which brings about false negative results. We have the invasive sampling to extract blood, and this is very painful, which makes it hard to get multiple tests. And also, there is need for expertise in operating these diagnostic tools, particularly the microscopes and the PCR machine, and also they are time-consuming.
Yes, more comparison of our proposed RDT and the methods of diagnosing malaria that has been there. The previous presenter said the microscopy, the PCR, and the conventional RDTs. We find that for the microscopy and the PCR, they always need access to electricity, expertise, they are non-user friendly, and they are non-field deployable, though they are accurate. Then for the conventional RDTs, the malaria detection is inaccurate because of the false negative results due to the histidine-rich protein gene deletions. So with our system that is molecular based, next slide, please. We see that synthetic biology-based diagnostics offer a real-time diagnostic, sensitive and highly and non-invasive technique for detecting the DNA of infectious agents. So our system is a cell-free modular detection system for people's sputum that uses a toehold mechanism to recognize the target DNA sequence. So a positive reaction on the paper would be seen after a short incubation as an appearing color. So to the user, it is affordable, it is relatively fast, and it doesn't require any specific laboratory equipment. It is shown in the next slide, just as you see the color on the last diagram there. So our objectives are generally, 1, to design and develop a rapid test to design and develop a rapid and non-invasive diagnostic test for Plasmodium falciparum, which causes malaria. And then specifically, we have to design and develop a toehold switch biosensor that specifically tests for the falciparum gene A in the saliva, and then also to design and construct a molecular CRISPR system mediating as a molecular converter, and then lastly to optimize the sensitivity and specificity for the falciparum gene A in saliva.
Okay, and then the process in which this is going to be done, we are going to implement the DBTL process, which is the design, build, test, and learn process. So in this process, there is the designing phase where it is going to be mostly in silico. So the in silico, we are talking of the modeling and also the designing of the CRISPR-Cas9 system using the CRISPR-Era tool, and also the toehold switch design and construction using the NUPAC. And lastly, now we do now the Golden Gate assembly of that. And then the building phase now includes now whereby the toehold switches will be generated using an original design algorithm. And then the testing phase is the performance of the design toehold switches, which will be estimated using computational modeling. And then lastly, now we do the iteration as we try to improve our system. So our system is a cell-free transcription and translation reaction, which is regulated with a toehold switch. So now the reaction is composed of an equal lysate, which provide translational and transcription machinery, and the toehold, plasmid, and trigger sequence, which are then the on and off switch and other necessary components such as amino acids, energy, reagents. So when the toehold switch is triggered with a target, it unfolds and it will start what the reaction expression of a reporter gene. So the reporter gene, we shall test the calorimetric reporter. So now the calorimetric reporter that we use, it's either it's the beta galactosidase, and then also we use the fluorescent reporters, which is the M-cherry. So now we'll be following the principles according to Beidels and Ramson et al. And then now concerning the toehold switch, now a toehold switch is a riboside, an RNA molecule that is looped molecule. So what it does is when a specific trigger binds the structure, it unwinds to reveal the start codon, allowing the translation to start as a ribosome binds to the ribosomal the RBS. So now this is how the concept goes. So the input is the Plasmodium falciparum double-stranded DNA. So now for the double-stranded DNA to become a single-stranded DNA, so we use CRISPR-Cas9 as the molecular converter. So it converts the double-stranded DNA to a single-stranded DNA. So we'll be targeting the STEVO gene sequence, which is found in the Plasmodium falciparum. It's a conserved sequence. So it will cleave on that. So it will be like a CRISPR-mediated strand displacement. CRISPR-mediated strand displacement. So it displays a strand so that it can be easily accessed by the toehold switch, because the toehold switch will only operate on a single strand. So using Cas9 as the molecular converter, and then the protease kinase will be the one now to separate that cleaved part from the CRISPR. And then it has to undergo pre-hybridization. So the pre-hybridization process is whereby the single-stranded DNA is also recuperated and recycled. And also we do what we call the reconstituted double-stranded DNA. And then on top of that, so when we've constructed the plasmids, so the plasmids now will be what? Will be transferred to the E. coli. So the E. coli that we're going to use will be engineered. We'll genetically engineer it so that we can have these two plasmids, the toehold plasmid construct and the CRISPR-Cas9 plasmid construct. And then it has to undergo cell lysis. So we'll lyse the cells and then do the lyophilization, whereby we have to freeze dry them so that we can make a cell-free system. We can put it on a clear disc of paper.
Okay, so the chassis selected here is E. coli K12. Why? Because it is one of the most intensively studied model organism in molecular and cell biology with a well-known knowledge of its genetic structure. It has a rapid doubling time of less than an hour, meaning that the saturated culture can be grown overnight. It has lost its ability to thrive in the intestine, so it is safe. That's a biosafety level 1 organism. Next slide. Now, in order to construct and assemble the gene, the Golden Gate cloning system will be selected as the assembly platform for generating the toehold sensor plasmid. We design our sensor plasmid so that the toehold switch is easily exchangeable with Golden Gate assembly, as the Golden Gate assembly is very flexible and efficient. Toehold sensor and CRISPR-Cas9 plasmid will be constructed. All assemblies will be designed and computationally modeled using the benchling or SNAP gene. The performance of the designed toehold switches and CRISPR-Cas system will be estimated using computational modeling. Next slide, please. Now, how are we going to improve our biosensor design? We will do that by amplifying the signal using the T7R NAP to improve the sensitivity and dynamic range. Equally, we will do a pre-hybridization reaction for the single-strand DNA enhancement. Now, how about the quality of our matrix biosensor design? We say it will detect low limits, or you have a high sensitivity to the DNA present in saliva, equally have wide dynamic range, and with a low leakiness.
So this technology has an advantage, which is just due to the complex when it comes to its design, which requires professionalism. And then onto the conclusion of all of it, we find that the innovation of this technology is a very big step in the field of diagnostics, biosensors, and also many other related applications. And we find that, as from other presenters, we find that this technology is able to be integrated with it's able to be integrated with various with the cell-free system and also the paper-based, and this makes it to be simple, to be easily portable, and also to be affordable. So in regards to this, it means that it can be used in detection in remote areas. Yes, and also we can see that it can be various detection platforms like fluorescent, colorimetric, luminescence, and also electrochemical outputs allows this technology to be used in diverse interdisciplinary applications. And as other presenters also talked of CRISPR-Cas9 technology, this use of this technology enhances the specificity, sensitivity of this technology. So in this project, we were a team of 5. A team of 5, that is Bukhosi, Leticia, Peninah, Eminem, and James. And we thank you for listening.